John Daniels. I'm at Oklahoma State University. And one of the things I wanted to bring up before I kind of go into my lecture is I'm part of a group here at Oklahoma State, which some of you might have heard of. We're called NIMFAB, but we're the National Institute for Microbial Forensics and Food and Agriculture Biosecurity. And the reason why I wanted to mention that, just to give you kind of a little bit of perspective, that our group approaches things from kind of the microbial forensics and biosecurity perspective, which is, I think, a little bit different than in some of the other sciences out there, and how they think about and go about their research. So, like, just one thing I wanted to talk about, because from my understanding, we kind of have a diverse group here, and not everyone might be a plant pathologist, but even if you're not, I think these are pretty easy concepts, especially if we think about this concept and relate to us as humans. You know, as a human, we have all kinds of organisms, microbial, fungal, and viral, and everything that aren't necessarily good for us. And on the plant realm of things, and agriculture, biosecurity, and these types of things, these same types of pathogens also have an effect on plants. But that doesn't mean that they're all bad. And so one of the challenges that we face is that, you know, a lot of these microbial organisms, like inside our gut, are very useful, and we need to have those. Now, in the plant system, it's the exact same story, where a lot of these microbial organisms are very useful for the plants. And so one of the challenges is, you know, who's a good guy and who's a bad guy? And can we go in and find the bad guy, you know, amongst all the good guys that are out there? And so that's one of the challenges that we face in plant pathology and diagnostics. Some other things to keep in mind is that, you know, there's a lot of things, you know, unfortunately, we haven't developed a plant that can talk to us. So until that happens, you know, we were faced with a challenge of, you know, is it actually a truly, you know, infected plant with a pathogen? And is it truly have a disease that's really presenting? You know, or is it just an abiotic factor like environmental stress, or perhaps a grower put too much nutrition down? And so what we think is actually a disease may not even be a disease at all. And this is a slide I always throw out there, and surprisingly, you know, I usually kind of ask people to give me their opinion on what they think it is. And so on this slide on the left that you're seeing here, I'm not going to ask anyone, but I've actually had a lot of even plant pathologists who try to guess, and I think they usually want to say some kind of fungal pathogen, but it's a lightning strike. And the one on the right is hail damage. And so, you know, if we were just to kind of glance down and see something like this, we might think it's disease, when in fact it's not even you know, a disease at all. It's an abiotic factor. And I'm not going to really talk too much about this, but I wanted to show you guys this slide again. I know that Tony brought it up in his presentation, but so the sequencing technology that we used was the 454 Junior sequencer. And really the main thing I want to point out is this last phase of the whole thing. So not so much as the technology that we use, but all the data that came out of this technology. And the reason why this is so important is because the data at this point in time, it's all stored as a digital value. So it's not actually like when you do PCR, we're not really working in that kind of environment. We're actually working in the computer environment, and that's really an important concept to understanding how the overall process works. And so why use next-gen sequencing? You know, the fact is, because as scientists, you know, more is better. You know, I think in human nature, more is better. And so the more information we get, why not go ahead and get it? But the problem is that, you know, a lot of times we're kind of left with, you know, what do we do with all this data? It's just too much. We don't know how to handle it, or my computer crashes when I try to do something, you know, and that kind of thing. So at least in diagnostics realm is, when we think about it, that not all that data is truly needed. And so if we're able to just kind of ignore the stuff that we don't really need to look at, we can really reduce the bioinformatic load. In a sense, that's kind of how the overall system works, and that's why we can run our system on like laptop computers that aren't these high-dollar machines and that sort of thing. Another important concept, and this is what I have found, at least in presenting this with my group, is that when you start talking about bioinformatics, everyone kind of, you know, at least from like a lot of plant pathologists, will sometimes be a little bit standoffish because they think it's going to get really crazy if the algorithms are going to start throwing at them or these types of things. And that's not really the case at all. But what is very important is that we're both speaking on the same terms. So we're like a bioinformatician, you know, what they might call like a query, someone on the plant pathology side of things, that could mean a totally different term to them. And so we would need to make sure that we're in sync with what we're saying and moving forward. And to kind of expand on that, I went ahead and threw up these definitions just so you guys have a bit of an understanding of what all we're talking about. And I know Tony he touched on this top one of the electronic probe, and I think he said it best, and really this is the best way to think about it. You know, it's pretty much, if you just think about it like a molecular probe, it's basically kind of doing the same thing. Now, later on in the afternoon, when you might see us talk about like a sample sequence database, that's just that's pretty much what we call the output file from an NGS run. Whatever the NGS came from, you know, whatever sequencer you use, we just refer to it as an. Now, these next two terms, a match and a hit, we kind of use those back and forth, but I just wanted to kind of lay it out there, and maybe I should have reversed this. Maybe if you look at it, the definition of a hit, which is just an association between a reference. The MSD, ignore that term, that should be NGS, so next generation sequencing read, and a query or an eProbe. So just an association between our eProbe and a read within an NGS run. And a match is basically can have one or more hits. So it's possible for a match to have, you know, up to like 20 hits on it. And then I think most of you probably have an understanding of what a read is from the next gen sequencing run. So before we go into a little bit more detail, I just want to kind of give I don't know how many of you have ever designed your own primers, but we actually do a primer workshop here. And when we do this, there's a whole slew of different programs that we use, and there's a whole bunch of different settings that we have to consider when we do this run, or when we design our primers. And so these are just kind of a list of some of the programs that we use in our primer design workshop. However, in saying that, within each one of these programs, there's other factors we got to consider, and I actually don't even have them all listed here. But GC content, the total length. You know, are you doing a multiplex? So you might need a little bit longer primer. And if you're just doing an endpoint PCR, your product size, your temperatures, and your thermodynamics, and your folding. You know, and so in a perfect scenario, if you've gotten all these things taken care of and you've developed your primers that you're going to use to look for something, there's still a chance that it may not work, and then you have to make sure you don't hit up on anything else, any other pathogen that's going to be close to it. There's a lot of time that's involved in this. So to compare this to what we're doing with our electronic probe design, just to hit a little bit on Tony's slide, you know, basically we take an entire genome of a pathogen, of a target pathogen, entire genome of something. The point I wanted to hit on again that Tony mentioned was this step right here where you have our final eProbe, this blast check. Because one of the questions comes up is, how do you guys really know, because you're using the whole genome, so how do you know that your probes aren't going to be hitting on other things? And one of the ways we know that we tried to remove that possibility is because after the initial phase of TOFI, after we go through and we kind of get these unique sequences, all of our probes, we'll blast those against the NCBI NT in our database, and then we'll go through and we'll use like a friend script is what I call it, and so the friend script will go in there and it looks for whatever you're assigning as a friend. So in my case, if I had like DC3000, I want you to keep it. If it hit on anything else, even if it hit on DC3000 plus other things, I don't want you to keep those. So just keep the things that hit on my target pathogen, which is DC3000. Now, I'm going to give you a little bit of the data that I've generated and how we went about doing it. And so what you're seeing here on this side, this is my target pathogen. So I have the Ralstonia silencerium, GMI 1000, and the DC3000. Over here is who I use as my near neighbors, and of course, you know, you can use these numbers to see what I pulled to generate my probes. And then along the bottom here, we have probe lengths 25 up to 80, and this is the total number of probes at each length. And this is for Ralstonia on this side over here. Okay, so to kind of help you get a little bit better visual on exactly how we went about the experimental phase of this. And so what we started with is, I grew up a healthy tomato plants. And I did basically total nucleic acid from healthy leaves, and you're seeing those leaves right here. So we had there was no disease present at all, and we did endpoint to verify there was nothing in there. So we did a total nucleic acid extraction. On the other side of it, I grew up the cultures of Pseudomonas DC3000, and then I did total nucleic acid extractions from those. And of course, when you bring the two together, and you have diseased plant tissue, which is what you're seeing right here. yeah, basically the same thing. We did a total nucleic acid extraction. So just to kind of show you the layout of this, so we have our starting material, which either was the culture, the diseased plant material, or the healthy tomato leaves. And we performed an extraction procedure to get the total nucleic acid. And then initially we kind of had different, I think 3 different protocols, which is basically what I'm showing here. So we had like a whole genome, a whole transcriptome, and then we also did a protocol where we had a combined of the 2 that were actually combined together. If they were separated, we would go back in and I'd just do a 50-50 via a 50-50 volume mixture based on volume, and then this would be sent off to sequencing. So where we are now is this little box right here is now right here. To kind of give you an idea of where we are on the experimental flow. So we've done the sequencing. We get our sequencing output file, which is all digital. It's a digital file, so it's an SSD. And now from my initial eProbe design, I have my eProbes right here. Those go in, we do the blast, and I think Tony's kind of talked about all these different, what happens after this phase here. So basically we are able to go in and say, yes, pathogens present or pathogens absent. So this slide right here is showing you the total output files from a 454 junior sequencing and the total numbers of everything. But really what I would like you to focus in on is this right here. So this is my barcoding sample. So I have an R11, R12, R11, R12. So the R11 is for DC3000, and for this one I have a total number of reads that came from this pathogen is 16,462, whereas 48,000 reads came from other things. My host and whatever endophytes were mixed in with my host. Now, when we run EDNA on this is basically the scenario that we saw happen. And so, right here, we're looking at the total number of matches along the left-hand column. Bottom here, we have our probe length, and then we have our various E values. And when we leave the barcode on, so the barcode wasn't removed in this case, we would call this as a negative, that the pathogen is not present. And keep in mind that I knew that I had 16,000 reads in there, and according to this, I mean, pretty much almost, I have no matches at all. And so the next thing we did is I went in, I cleaned it up, and I actually took the barcode off. And so by taking that barcode off, when I went from like 2 to 3 matches, I'm going up to 243 matches at the length of 25, up to 3,000 matches at 40. And then at 80, I have around 1,000 matches. And keep in mind that these are pretty stringent values. You're up to 10 to the minus 9, so your stringency is going way up. You're still getting a high number of matches. So this is the number we would expect to see. This is what we would not want to see. So what this tells us is the importance to removing the barcode, or I know some of these sequencers, they'll add like a primer. So you need to make sure that primer gets taken off there and your sequence is cleaned up just a little bit. Now, moving out to the field, when we look at our field data, and where we combine the EDNA and an NGS run, and so the tomato plants that were used out in the field are the ones that you saw in the image, actually diseased. So we had great disease symptoms, and basically what we're seeing is that we can call this sample as a positive call for our field samples, which is typically what you'd probably expect to see if you have something coming into your ports, or if you have a grower who's growing something, you have disease symptoms. So these are the numbers that we would expect to see. But then something interesting also happened, and that I used. So on this one, sorry, I went back to the previous slide. So you're looking here. This is the DC3000 eProbes. Now I also generate eProbes for the Ralstonia. This is where Ralstonia came into play, and this is for the just GMI 1000 eProbes. And so the same sample from the previous, this data right here. So you have the 454 run of symptomatic tomato plants. You have the exact same sample here, but with the Ralstonia eProbes is now searching this, and in fact we have positive hits on the sample using the Ralstonia eProbes. And to verify this, I had sample material that was sent to Dr. Schneider's lab at the USDA ARS group, and they verified that in fact there was a Ralstonia infection in this plant. It goes to show that EDNA is not only capable of finding a single pathogen, but we actually can find multiple pathogens that basically we have a co-infection situation going on here, and we were able to go in and see both pathogens. and pull both pathogens out of this infected plant material. Okay, so I'm just showing you here just kind of the total number of the Ralstonia E probes here to give you an idea on how many of those probes that